Hello everyone. Very good morning to all of you. Myself Dr. Rajib Deb. I am working as postdoctoral research fellow at Animal Biotechnology Center, ICAR-National Dairy Research Institute, Karnal, Haryana, India. Okay, we'll be presenting on the topic of lateral flow immunoassay for rapid detection of Staphylococcus species in cow milk. So let us have brief introductions. As we know that Staphylococcus species is one of the important common foodborne zoonotic disease in human, and thus identifying those bacteria in food samples, it is a important legal testing item. And thus, in order to ensure the food safety, it is necessary to develop particularly quick and precise identification method for Staphylococcus species in dairy milk or mastitis milk sample. So far, there are different methods are available. Like first of all, it is that conventional method, that is the culture-based method. But the disadvantage of this culture-based method is that it is time-consuming. It may take more than 2 days. Then it is tedious. Further, it requires extra care for specific detection of particular organism. Second, it is certain immunological method like ELISA, then Western blotting, immunoprecipitation test, and all. But these assays having lower sensitivity and they are complex, tedious in operations. Thus, it is hard to popularize to these methodologies. And nowadays, mostly PCR-based methodologies are also using for detection of defined kinds of food pathogens. Although PCR-based method is very much specific, sensitive, and it is widely acceptable methods. However, it requires certain sophisticated instrument. For example, your thermocycler, then you require gel documentation system, then agarose gel electrophoresis apparatus, etc. So there are some drawbacks of this, whatever the conventional methodology so far available. So in this work, we have developed a lateral flow assay for detection of Staphylococcus species in cow milk. For developing this lateral flow assay, we have targeted specific antigen of Staphylococcus aureus, and we have gone for raising hyperimmune sera in rabbit. Followed by, we try to synthesize the nanoparticles from mango leaves. As we know that the nanoparticles, it is can be originated from different origin, like it may be gold nanoparticle, carbon nanoparticle, or maybe silver nanoparticle. And these mango derived silver nanoparticle, they are the green nanoparticles, and they are non-toxic, and as it is easily available, so we chosen these mango leaves for synthesizing the nanoparticle. Followed by, we have gone for their characterization by UV light scanning, Zeta analyzer, FTIR, etc. Further, we have gone for development of lateral flow immunoassay. So this is the serological assessment of the polyclonal sera, which is raised against the Staphylococcus aureus specific antigen in rabbit. If you see this first graph, it is showing the testing of the pre-immune sera, and we have diluted the sera, and we have seen that there is no titer it was observed. So it indicates that the rabbits, they were free from any of the Staphylococcus infection. Coming to the figure number B. In figure B, we have collected here the sera from defined days interval from 7, 14, 21, 28, and 35 days. Then we have gone for it is serial dilution. And we have seen that at 1:300 dilution at 35 days, we are getting the highest titer. So we have chosen the 1:300 dilution at 35 days sera to detect the limit detection of the antigen, and we have seen that it can detect up to 0.5 nanogram per ml of antigen in the milk. Followed by, we have gone for the synthesis of silver nanoparticle from aqueous extracts of mango leaves. For this purpose, the mango leaves, we have choked it, then we have taken that aqueous leaf extract and we have mixed with freshly prepared silver nitrate. And this mixture then further 20-minute incubation and sonication for 10 minutes. we got that probable silver elemental nanoparticles, which we have seen by UV light scanning that it is giving the highest peak at 442 nanometer. So followed by, we have gone for some sort of characterization of this nanoparticle. We have gone for zeta Sager analysis by DLS or dynamic light scattering, and we have seen the size of the nanoparticle, it is 78 nanometer. Followed by, we have gone for Fourier transform infrared spectroscopy or FTIR analysis for the both extract and nanoparticle. If you see here in this extract, we have shown three stretches at primary amine stretch, second is saturated hydrocarbon or carbonyl group stretch, and third it is O stretch. So this indicate that this extract having certain phenolic compounds. But once we seen that the elemental nanoparticles that FTIR has been conducted, here if you see this peak, it is missing. That is, there is no saturated hydrocarbon or carbonyl groups. So it indicates that this might be possible that this saturated hydrocarbon or carbonyl group has been used for reduction of this silver nitrate to the elemental silver. Thus, and moreover, these hydrocarbon, they are toxic materials. So it also indication that these green nanoparticles, they are harmless or they are non-toxic. And further, this nanoparticle we have synthesized as powder form, and we have seen that this powder form can be stored at room temperature for a long time, and it can be used at any time. So followed by, we have gone for conjugation of those silver nanoparticle with antibody. Conjugation, it can be done by active or passive methods also. We prefer here passive conjugation because in passive conjugation we require less antibody and it is more stable compared to the active conjugation. So we have standardized this passive conjugation method with defined titration buffer, and we have seen after addition of the 10% sodium chloride in the conjugated mixture, the color remain similar. So we have chosen the pH is better here, where we got the right shifting of spectrum from 442 nanometer to the 448 nanometer. So this is the schematic representation of the developed lateral flow assay. So we tried for both competitive and sandwich based lateral flow immunoassay. In sandwich, I have not shown here. This is as I think all of us, we are knowing about the lateral flow strip, or just simply briefly I am describing it here. A lateral flow strip having defined components. One is sample pad, second followed by conjugated pad, followed by the test line, control line, and lastly it is the absorbent pad. Sample pad is the one where we load the sample, and conjugate pad is the one where we will load the conjugated antibody with the nanoparticles. And test line, it is your own choice. Here you can put antigen or antibody. So we tried both antigen and antibody. Antibody, if we try, this one become a model with sandwich. And if we put the antigen here, it is model of competitive lateral flow. And control line, we have imprinted with the anti-rabbit IgG. So this is the advantage of this competitive lateral flow, that we don't require to put the antibody, and moreover antibody, it is costly to synthesize and all, so it is reduce the cost of the assay. So here the samples for getting more specificity, we have standardized the method. The milk sample we have enriched with 10% substrate A and pinch of chromogenic dye, as it is some sort of this copyright issue, so I am not mentioning here what is the substrate A. So after that, the red color form, followed by we have gone for 6 to 7 hour incubation at 37 degrees centigrade, and once the color get yellows, that we loaded in the sample pad. So if it is the positive for step, we will get line at control line and test line. Where it is negative, then we will get only control line. So these are certain some kind of optimization of the test line developed for the lateral flow strip with several level of primary antibody concentration, which is loaded on the nanoparticle conjugated suspension. And if you see here, up to 5 microgram per ml antibody, which has been conjugated with the nanoparticle, also we are getting the test line. but to get better result, we chosen the 10 microgram per ml of antibody, which will be conjugated to the nanoparticle. In that B, this is the what is the concentration of antigen we loaded to the test line. So we have seen that up to 0.5 microgram also we are getting means visible band, but we have chosen the 1.5 microgram for better visibility. So now coming to the sensitivity of the assay. So as we know that any of the assay developed, the sensitivity and specificity are more important factors for a successful assay. So the sensitivity we tested, and it was detected that we can detect up to 1000 bacteria per ml, that is 10 to the power 3 CFU per ml we can detect. And we have seen this is similar to the sensitivity of the conventional PCR based methods. Followed by, we have seen the specificity of the assay. We have targeted different kinds of microbes like Staph aureus, E. coli, Streptococcus achalaxia, Pseudomonas aeruginosa, and Acinetobacter johnsi. And we have seen that it is only given the control and test line for Staph aureus, whereas control line can be formed in any of the pathogens. So followed by, we have gone for validation of the assay. We have used the reference culture methods to compare with our developed lateral flow in assay, and we have tested I think 154 samples here, and we have seen the relative accuracy of the assay, it is 97%, relative specificity it is 98%, and relative sensitivity of the assay is 96%. So with this, I would like to conclude that lateral flow assay can be an alternative to the conventional method to develop means specific and sensitive as well as user-friendly assay for detecting the Staphylococcus species in milk samples, and it can be used as pan-side diagnostics. So thank you.